So, ladies and gentlemen, welcome to this afternoon's event. We're at the beginning of a new academic year, and we've got really quite a panoply of stars speaking to us over the next 13 weeks through the Global Institute of Water Security Distinguished Lecture Series and a few extra friends. It's very nice to start this series off with a fellow Canadian. There was, as many of you know, a competition by the federal government to bring in distinguished scientists to Canada, appointing them to a Canada Excellence Research Chair. And Philippe and I were two of the initial batch of 19 and the two key people in water, and it's probably appropriate that we represent the two leading institutions in water in Canada, arguably the Saskatoon and Waterloo. Philippe's title is Canada Excellence Research Chair in Ecohydrology and Biogeochemistry, and he's been doing some very exciting work on biogeochemical processes associated with soils and rivers, and particularly surface water, groundwater interactions and riparian habitats and the like. And he works across a range of scales which run from soil columns to large river systems to global river systems. And he's going to talk to us about a flavor of that work today. So Philippe, like me, is a transatlantic person. He began with his first 2 degrees at the Free University of Brussels. He migrated to the States for a PhD at Yale. He's had appointments at Utrecht in the Netherlands, and then more recently at Georgia Tech, where he had a title of eminent scholar, I think. He's, for those of you publishing in the hydrological literature, he's been involved at a very senior level with the major journals. He was co-editor-in-chief of Journal of Hydrology, for example. So he's an important person to keep in with. Anyway, he's here for a few days. He's been meeting many of you. The idea is that we look for synergies in our research programs at the 2 universities and for some opportunities for future collaboration. He's already been working with us on some of the Diefenbaker work. So it's really a very great pleasure to welcome Philippe. He's truly a very eminent scholar, and I'm really excited to look forward to his talk on environmental and biogeochemical effects of river damming. Philippe, thank you very much.
Thanks. Very nice. So, good afternoon, everyone. So, first of all, it's a pleasure to be here. It's my first visit to Saskatoon and the University of Saskatchewan. And so I certainly would like to thank Howard for the invitation and making this visit possible. I'm here with 2 colleagues. Feridun and Taylor are 2 colleagues in my group, which are also very excited about visiting the university here and the Global Institute for Water Security. So we had a very full day. We started sometime around 9 o'clock. We've already talked to a lot of people. We've visited some of the facilities, and so far it's been very exciting, very rewarding. So I'm looking forward to the next two days. So what I would like to do today is talk a little bit about environmental and biogeochemical effects of building of dams on rivers. And I'll focus on the effect that dams have on the fate and behavior of nutrient elements in river systems. Get this right. So first of all, I'd like to acknowledge a few people that in my group are working on this particular subject. So Taylor, who is a PhD student and whose work I will be presenting largely today. Severin is another PhD student who is working on environmental effects or algal blooms in the Three Gorges Reservoir. I'll touch on that very briefly. And then Jennifer Hood and Hans Duur. So I don't think I need to convince anyone here about the importance of rivers and streams and the associated riparian areas and floodplains, but I'll go to this list anyways. Rivers do provide a lot of very important services. In particular, they act as the main connectors within the freshwater cycle. So they connect the different parts of the freshwater cycle. They're of course very important for water supply, but they also have a lot of non-consumptive functions related to transport, food production, and energy generation. They contribute to water purification, to waste assimilation, and erosion control. In very many cultures, they also are part of the historical heritage of a variety of societies. From a more ecological viewpoint, they of course offer a lot of different habitats for organisms. And what really interests me is that if you look at the water quality or the composition, the chemical composition of a river, it actually integrates a variety of, essentially it's an integrator of all the physical, hydrological, biogeochemical processes that take place in the upstream watershed. And then, and I'll also touch on that today, of course they're very important conduits for the transfer of materials from land to the ocean. So they also connect the terrestrial realm to the oceanic realm. But of course, there's the rivers, or the health of rivers, is also threatened by a variety of different anthropogenic pressures. For instance, excessive water abstraction, channelization, urbanization, eutrophication, damming, the release of pollutants, habitat fragmentation, which is an important subject of a lot of research, and actually very exciting research using, for instance, genetic techniques, invasive species, and of course, also they can be threatened by climate change. So today I'll be focusing on the two aspects that are highlighted here, eutrophication and damming, and also the interactions between those two. And so again, I'm sure that most people are aware of this, but one of the most important anthropogenic pressures on the environment is the fact that we're releasing more and more nutrient elements to the environment, and ultimately these nutrients might make their way to river systems. And that's illustrated here for the total human nitrogen input to the environment as a function of time. And you see it after 1950, because of increased use of fertilizers, the nitrogen loading to the environment dramatically increased. And similar stories also pertain to phosphorus, sulfur, potassium, etc. So what I call here the anthropogenic nutrients. And of course, these nutrients ultimately make into the river system, and they might have significant impacts, ecological and environmental impacts. And in particular, when we add nutrients to an ecosystem, in particular to an aquatic ecosystem such as a river, we will enhance primary production. And so what increased nutrient supply to rivers can do is to offset or change the balance between production and respiration. And this then, in turn, will affect the exchanges of gases between the river and the atmosphere, in particular CO2 and oxygen. Now, if you take most rivers in their pristine state, and there's not many rivers left in their pristine state, but if you can reconstruct their pristine state, usually respiration dominates over production. And as a result, and that's what we call, therefore we call this state heterotrophic. That also means that the river is a net source of CO2 to the atmosphere. And what often happens when we start adding things like nitrogen and phosphorus to these rivers is that we shift the balance so that ultimately production might actually start dominating over respiration. And at that point, the river becomes a net sink for CO2. And that actually has been documented for a few rivers in the world. Here what I'm showing is data where these are all the data, the points that you see on the graph are reconstructed partial pressures of CO2 in water of the Yangtze River, and it's in a station, a downstream station in the coastal plain. And if you go back to the 1960s, or the mid-1960s, you see that the waters were supersaturated with CO2. In other words, the river was degassing CO2 to the atmosphere. But what you see as a function of time is that we have a decreasing trend in the supersaturation, and by the turn of the century, in fact, the water is very, very close to saturation. In other words, there was no net exchange of CO2 between the river and the atmosphere. And if you then extrapolate this trend, then by now, the Yangtze River, at least the downstream reaches of the Yangtze River, have become a net sink for CO2. So they've changed the role of the rivers, or at least this particular river, or this particular part of the Yangtze River, has changed, its role in the carbon cycle has changed. And this is probably affecting, these type of large-scale changes are probably occurring in many rivers on the world. Of course, some of the nutrients are then transferred by rivers to the receiving water bodies, where they can cause things like algal bloom, so excessive algal growth. Here's a picture, for instance, showing the 2012 Lake Erie algal bloom. in the western part of Lake Erie, and this year we had another algal bloom occurring. Now, the exact mechanism of these algal blooms is not really understood yet, but ultimately it's because nutrients, or excessive or extra anthropogenic nutrients, have been added to the system. And here, for instance, is a nice picture of a bloom that took place last year along the coast of China in Qingdao. And according to my Chinese colleagues, these type of algal blooms are occurring with increased frequency and increased intensity. All right, so there's a lot of interest in trying to understand and manage the occurrence of these blooms. But when we think about, or when we talk, or when we consider nutrient loadings to river systems or to ecosystems in general, it's not sufficient just to think about the absolute flux of added nitrogen or added phosphorus or added other nutrient element. It's also important to look at the ratios between the nutrient loadings. And so again, if we go to the Yangtze River, here what's plotted is, over time, the changes in the silicon to nitrogen ratio in the water of the Yangtze, in two stations in the upper part of the Yangtze, in the middle part, and in the lower part. And if you first look at the top two panels, in the upper part of the watershed, you see that essentially over time there's very little trend in this silicon to nitrogen ratio. And that's because essentially there's not too much human activity taking place in the upper part. But if you look in the middle and the lower part of the river, you see a dramatic decrease of the silicon to nitrogen ratio over time. And the reason for that is that in these parts of the catchment, more and more fertilizers are being used, there's more wastewater discharge that brings more nitrogen into the system, but the supply of silicon has remained more or less constant. Most silicon actually comes from the weathering of rocks, and that is less affected by anthropogenic pressures, it's less affected by human activity. So if the silicon supply remains more or less constant, the nitrogen supply goes up, the silicon to nitrogen ratio will go down. And what that means is that over time, it's going to become more favorable for organisms that don't need silicon to compete or to outcompete organisms that do need silicon. So I want to say a few things about silicon because typically it gets much less attention than nitrogen and phosphorus. Now, nitrogen and phosphorus are, I would say, universal nutrient element because all organisms need nitrogen and phosphorus. Not all organisms need silicon. But if you do what we call a supply and demand analysis, if you take a particular nutrient element and you look how much in a given ecosystem is needed to sustain the amount of primary production in that environment, and then you compare that to how much of that element can actually be supplied by the environment, so you do a supply-demand analysis. If you do that on a global scale, so if you do it for the entire biosphere, this is the order of elements that you get. So this is for the entire biosphere, terrestrial and marine included. What you find is that the first element to appear there is phosphorus, which means that it's the element in the shortest supply relative to the demand. In other words, it's the most limiting element, globally speaking. Next comes nitrogen, and about similar for iron. Now, for iron, it's mostly the marine environment that can be iron limited, right? So it's not so much of an issue on land. And the next element is silicon, right? So silicon is actually more limiting for the global biosphere than elements such as carbon or sulfur or potassium, elements that we typically associate with life, right? So it is actually a very important nutrient element, and certainly for certain organisms, it can be a limiting nutrient. Now, the reason why organisms use silicon is to produce structural elements made out of silica, SiO2, and these are used for support or for protection. So here I've got some pictures. I hope you can see those in the right-hand corner there. These are structural elements made out of silica that are produced by horsetail, which is actually a known silicon accumulating plant. And what's particularly interesting about these deposits is that they have extraordinary mechanical strength. They're some of the strongest natural materials that you can find. Yet they're actually very porous. So it's a very strong material, but it's actually also a very light material. And of course, that's what you want for support. You want something that holds you up, but doesn't weigh you down, right? So it's actually what a lot of plants use to give themselves rigidity or strength. A good example for that are grasses. Now, that's for terrestrial plants. If you go to the aquatic environment, then one of the main groups of siliceous organisms are these diatoms. These are unicellular photosynthetic organisms, and they make these little houses, these little frustrals out of pure silica. And as you can see, they come in a variety of different shapes and forms, and of course that's great if you're a diatom taxonomist. But the important thing here is that these organisms cannot survive, or they cannot thrive, they cannot prosper if they don't have enough, they don't have access to silicon. Okay, so now this brings me to river dams. In the early years of this century, a number of papers were produced highlighting the fact that river dams can actually lead to silicon retention. This is often referred to as the artificial lake effect. The idea here is that silicon that usually or before damming, just made it down to the lake or the coastal zone where it could be used by, for instance, diatoms, now is going to be retained by the dam. And a very influential paper was this one by Christoph Humborg, and you can see the title is Decreased Silica Land Sea Fluxes Through Damming in the Baltic Sea Catchment. And so what these authors propose is that the observed decline in diatom populations in the Baltic was actually due to the fact that after the Second World War, a lot of dams were built, particularly in Sweden. Sweden wanted to boost its hydroelectricity production. But one of the unintended side effects of this dam building was that the diatom population started to decrease because the availability of silicon in the Baltic was decreasing. And that's particularly visible in, so this is data of dissolved silica versus time from the 1950s to the year 2000. And if you look at the lowest set of data, it's this one here, that's the dissolved silica concentration in the upper part of the water column, so the photic zone where the diatoms actually have to live because they're photosynthetic algae. But even in the deeper parts of the Baltic, you see this decrease, right? So this would be an example of a regional effect of river damming or a downstream and an unintended effect of river damming on the Baltic ecosystem. So before we go further into, I'll delve a little bit deeper into that. Obviously, people have been building dams for a very, very long time. In fact, since agriculture was started, people started damming rivers to divert water for their crops. But river damming, or intensive river damming, really took off after around the 1950s. It peaked in around the 1970s, and it's now slowing down, but it's still continuing, particularly in some of the emerging economies of Southeast Asia and South America. So we're damming. And of course, there's a lot of good reasons why we would dam rivers. First of all, they help protect us from floods. They stabilize water supply. In fact, it's estimated that between 12 to 16% of the world's food production is directly contributed through water or irrigation water from dam reservoirs. And 19% of the global electricity production is hydropower. So it's very important. And of course, also sometimes used for recreation. But there's quite a lot of potential ecological impacts, both at the local, the regional, and the global impacts. So I already gave you an example of the regional impact with the Baltic Sea, for instance. We'll look a little bit more at local and global impacts in a few minutes. So just to give you a couple other facts about dams. Worldwide, there are over 75,000 dams. So, and this number keeps increasing. So nowadays, more water is stored behind large dams than in natural rivers, all the natural rivers together. Dams intercept more than half of the natural freshwater flow from the continents to the oceans. So we're essentially starting to short-circuit the hydrological cycle. And there's, of course, some social costs associated with dam construction, and it's estimated that between 40 to 80 million people have been displaced from their homes as a result of dam construction. But the bottom line is that river damming represents one of the major human-induced modifications of the hydrological cycle in general and of river systems in particular. All right, so the hydrological impacts of damming a river are relatively well known. This is a very famous graph that you can get from the internet, from Wikipedia, and it shows the monthly average discharge of the Nile River before and after the construction or the closure of the High Aswan Dam, the Aswan High Dam. And of course, what you see is that the very strong seasonality in river flow completely is eliminated by the dam. So, and of course, this peak flow here is the famous flow that produced the famous Nile flooding events. All right? But what I really want to talk about today is how does damming affect nutrient fluxes in rivers? So when you build a dam on a river, essentially what you do is you take a stretch of a river that essentially before is just a conduit to transfer nutrients from upstream to downstream. into something that's more like a lake. And what's happened is that the flow velocity will slow down, the sediment will sink out of the water column, and because of the increased residence time and the decreased turbidity, primary production starts taking place in the reservoir. So we set into motion the carbon and the nutrient cycles within the reservoir, and this can also lead to retention of some of that carbon and some of these nutrient elements, nitrogen, phosphorus, silicon, iron, potassium, etc. So what we have here is that we can have, of course, ecological impacts within the reservoir itself, so in-reservoir effects, but we also have downstream effects because we're retaining some of the nutrients in the reservoir. And so, of course, a very important question is not just how much phosphorus or nitrogen or silicon am I retaining by constructing a dam, but also how am I changing the ratios of these nutrients that are transferred downstream beyond the dam. All right, so to show you some of the research projects we're involved in, first of all, let's talk about some of the in-reservoir processes. So we're working together with some Chinese colleagues on the Three Gorges Reservoir. So as you probably know, the Three Gorges Reservoir is on the Yangtze River. It's one of the biggest dams in the world. It's about 2.5 kilometers across. And what is also special about the Three Gorges Reservoir is the very, very large variability in water level. So there's about a 30-meter difference between the winter high stand and the summer low stand. So we just started, well, this project is still in initial stages, so I won't show you too many results. It's a very exciting project, so I want to talk a little bit about it. So if you look at the reservoir itself, it's over 600 kilometers long. It covers an area of over 1,000 square kilometers. And what's also important for the Three Gorges Reservoir, there's about 40 large tributaries. And so here's one of the tributaries we've been doing some work on. And the reason why we focus on the tributaries is because the major environmental impacts of the Three Gorges Reservoir are actually observed in the tributaries, not in the main channel. And one of those effects is quite important algal blooms. They recur not every year, but on a very frequent basis. And here, for instance, are two pictures that show the development of a dinoflagellate bloom. And in the picture up here, the main reservoir, the main channel of the reservoir with somewhere in the background there. All right, so one of the things that is very clear from the data that we've been able to assemble so far is that these algal blooms are very strongly linked to the hydrodynamics in these tributaries. And for instance, here you see on the upper graph here that between mid-February and mid-March, the water column goes from being completely well mixed to being completely stratified. And at the same time, this coincides with the massive growth of algae in the surface layer. Now, that's not totally unexpected. It's also known as the critical depth phenomena, is that as you stratify the water column, you can find the phytoplankton in the very top photic zone. And if there's sufficient nutrients available, then the algae will grow. So far, what we have been able to establish is that, first of all, these tributaries are high nutrient environments. They receive a lot of nutrients. So when we started off, the idea I had is that, well, these nutrients come from the catchment of the tributary, because there's agriculture taking place, there's urban development, so there's certainly nutrients coming from the catchments. But in fact, what has come out of this research is that most of the nutrients delivered to these tributaries actually come from the main channel. There's, of course, very complex hydrodynamics. Here, for instance, is for one of particular time during the year in the Changqing tributary, the representation of hydrodynamics. So the colors stand for the temperature. And the arrows, if you can see them, show the flow field. And one of the things that you can see, this blue mass here is essentially water that is coming from the main channel, carrying nutrients and upwelling into this tributary. And so at the surface, you then have a net flow towards the main channel. So these systems are actually, by our Chinese colleagues, refers to as estuaries, because this is a classic estuary in circulation. But it's the main channel that is bringing in the nutrients. And what is also very clear from the observation is that stratification controls both the onset and termination of the algal blooms. So what we've started to do, and again, it's very preliminary, so Severin, one of our PhD students, has started to basically develop models that couple the hydrodynamics to the plankton dynamics. For the moment, he's been running these simulations in very, very simplified model environments, but the ultimate goal is to go to more realistic model environments and see if we can actually reproduce the timing and the spreading of the algal blooms in the tributaries. The algal blooms tend to be mostly confined to the back part, the upstream part of the tributary. Okay, let me now switch a little bit and talk about how the building of dams is affecting what goes through the dam. In other words, how does damming affect riverine nutrient fluxes? And so I'll start by illustrating this with silicon. Again, we use the nutrient silicon. This is work that we're also starting to apply to phosphorus for the moment. A very simple question. In fact, this is the question I posed to Taylor for her PhD research. Tell me how much nutrient silicon is returned by river dams. Now, usually when a PhD student comes and asks, do you have a research topic for me? You start with a very complicated explanation. Yeah, very simple question, right? Now, sometimes simple questions don't necessarily have simple answers. So what you can do is, of course, you can answer this question, or try to answer this question for an individual reservoir. So that's where Taylor started collaborating with people from the University of Saskatchewan on Lake Diefenbaker. And so she started looking at data for sediment traps and sediment cores. She measured actually the reactive particulate silicon, trying to estimate then how much accumulation of this reactive particulate silicon is taking place through sedimentation inside the reservoir. And so if you know how much reactive silicon is coming into the reservoir, if you know how much accumulates, you also know how much goes through, at least on a yearly averaged basis. So she came up with this budget here. But the important point is that if you look at the retention of reactive silicon in this reservoir, it's between 73 to 77%. So the majority of the silicon that enters the reservoir stays in the reservoir, is not transferred to the downstream reaches of the river. So keep this in mind, this kind of 70-ish number. All right, so this is for a single reservoir. But now the next question is, well, can we extrapolate this? Can we scale this up to the global scale? So the next question was, well, can we do this at the global scale? Well, so what Taylor then did was he went to the literature and said, okay, let's find all the reservoirs for which I can establish, there's enough data to establish a silicon budget, so for which I can calculate silicon retention. Fortunately, she only found 17 reservoirs, and she did a very exhaustive literature search. So that only represents, so 17 reservoirs is less than 0.03% of all reservoirs worldwide. So there is no chance in the world that the 17 reservoirs are actually representative of all reservoirs in the world. So how do we go about? Do we just take the average retention for these 17 reservoirs, multiply it by the surface area of all reservoirs, and then come up with a number? Well, there's probably a huge uncertainty, an unquantifiable uncertainty associated with that. So is there a way around? Well, the way we decide to go around is to say, well, we actually know quite a lot about what happens to silicon when it enters a reservoir. So if you have a supply of silicon to a reservoir under dissolved form or particulate form, the dissolved form can be taken up by siliceous organisms, right? Mostly diatoms. So the diatoms or other siliceous organisms take up the silicon, they make these silica deposits. The silica deposits can then redissolve, but ultimately some of it will accumulate in the sediments. So what you can see here is a very simple mass balance model that we can establish. It's a very generic, it's not very detailed, it doesn't apply to any specific reservoir in part, but it represents overall the dynamics of silicon that we expect could take place in a reservoir. So uptake by siliceous organisms, recycling or regeneration of the silicon, and some retention in the sediments. So if we can take this model, then we can use our 17 reservoirs for which we have data on silicon budget to calibrate and verify that model. And if we could then take that model and apply it to all the dams or all the reservoirs in the world, then we could make a meaningful extrapolation from our 17 reservoirs to the global scale. In fact, luckily there is this database here, the Global Reservoir and Dam Database, where approximately more than 70% of all the dams in the world are in the database. You won't find anything on nutrient retention in that database, but you will find some basic information on the reservoirs, how big they are, how much water flows in, so the water residence time you can calculate from that. Some basic hydraulic information is available for all these reservoirs. So the approach we then use is to say, well, let's start with our 17 reservoirs. So our 17 reservoirs for which we have information. We can use that data we have through statistical analysis to figure out what are the key properties and forcings, or key properties of the reservoirs and forcings on the reservoirs that will affect silica retention. Let's merge that with what we know about silicon biogeochemistry. Let's build our mechanistic biogeochemical model. And then the next step is to say, well, if you have a model, typically you have a whole bunch of parameters. Can we come up with reasonable ranges for these parameters? Well, for the parameters that pertain to the reservoir themselves, we can go to our GrandD database and come up with reasonable ranges for those parameters. For the parameters that directly link to the silicon biogeochemistry, like for instance the uptake efficiency of silicon by diatoms, the dissolution rates of biogenic silica once it's released from the organisms, we can do that to then come up with ranges for these parameters. And then we use a technique called Monte Carlo simulations, where we basically run our mechanistic model over and over and over again with all possible sets of parameter values. And this then allows us to derive predictive global relationship that will relate the silicon cycling or the silicon retention to known reservoir parameters from which we can then extrapolate or estimate the global silicon retention by river damming. But the basic idea is that we start with a sparse data set, but we compensate the lack of data with what we actually know about the mechanisms, about the basic biogeochemistry of silicon in reservoirs. So here's, for instance, you can, of course, then, since you have a model, you want to have some level of verification. With these models, you can never have 100% verification. But one of the major parameters in our biogeochemical model is, of course, how much biosiliceous productivity can be taking place in a reservoir. So what you see here is when we fit our mechanistic model to our 17 reservoirs for which we have data, we get these dots here, these points. So that are the model predicted siliceous productivity. It's actually the maximum. So it's the carrying capacity of the reservoir to support siliceous organisms. And what we find, and this is not a correlation, this is what we find, is that these siliceous productivities actually correlate very nicely with the input of reactive silicon to the reservoir. And that is very satisfying because that makes complete sense, right? The more silica you supply to the reservoir, the more siliceous productivity, the more the siliceous organisms can thrive in that reservoir. So this is an outcome of this model. So that tells us that our model, although it's very simple, actually carries some reasonable dynamics in it. There's something that is correct about the model. So then we go to our parameter ranges. I won't go into detail here, but you can see that we do consider fairly large ranges in most parameters. So for instance, you look at the volume. If you look at the GrandD database, so this database, this global database for reservoirs and dams, I mean, we have reservoirs of about 1,000 of cubic kilometers all the way to 180 cubic kilometers, right? So what we then do is a Monte Carlo simulation. So what we do is basically we randomly choose parameter values. We run the model with that set of parameter values. We do this over and over again. I think Taylor did this for 6,000 times. And so what you can then try to do, and that's what we're attempting to do with this, is then to relate what we want to know, the silicon retention, for which we have little data, with some properties of the reservoirs that we do know. And for instance, what we do know from our database is the residence time of the water in the reservoir. And so if you take any of these, the data here are binned, but if you take, for instance, these guys here, those are the reservoirs that have water residence time between half a year to 1 year. If you run your model 6,000 times, you will find a very large range of silicon retentions, essentially going to all the way from about 0 to about 1, so from no retention to 100% retention. But most of your data will actually fall in a fairly restricted set of values, where the lines here represent the mean and the median. So the idea here is that if you go out there and randomly select a reservoir, and say that of a reservoir which has a residence time, a water residence time between half a year and 1 year, it's more likely that the silicon retention will be somewhere close to the mean and the median than it will be close to one of the extremes. So that's how we get these global predictive relationships. And so then you apply this to the entire database that you have, and this is then the number that Taylor came up with, right? So number doesn't mean necessarily very much. 22.3 teragrams of SiO2 per year. Tera stands for 10 to the 12. So it's a big number, right? But when you do these global estimates, that's what you get. You get big numbers. So to make it a little bit more palatable, if you take the mass, this mass of SiO2, so this 22.3 teragrams of SiO2, it's equivalent to the weight of 66,000 Boeing 747, right? And why is that important? Well, why is that more palatable? Well, remember, we have about 70,000 reservoirs. We have about 66,000 of these big airplanes. So on average, in each reservoir, each year, one of these big airplanes full of silica is getting retained in the reservoir. So it gives you some kind of an idea of how important, what kind of a masses we're actually talking about. Now, if you look at the total global silicon loading to rivers, it represents about 5%. So 5% doesn't sound like a lot. But remember, we keep building more and more reservoirs. In fact, we keep building more and more really big reservoirs with lots of water residence time, which tend to retain a lot of silicon. So in the future, this percentage is going to go up. In addition, remember that the silicon loading does not vary very much over time, but we keep increasing the loading of phosphorus and nitrogen, so which will push downstream systems more and more towards silicon limitation relative to nitrogen and phosphorus. All right. The other thing is, because we use a mechanistic model, we can also make predictions. For instance, this is then from all these 6,000 model simulations, what we get in terms of the distribution of silicon retention times. So first of all, you can see that the average silicon retention is 20%. So 20% of the reactive silicon coming into the reservoir is being retained, and it's skewed towards the lower retention times. So remember now Lake Diefenbaker. Lake Diefenbaker, we found about 73 to 76% retention. So that's out here. So in that respect, Lake Diefenbaker is kind of an outlier. But that result is also important, because now you can ask the question, why would a reservoir like the Diefenbaker be so very different from the average reservoir on the world? So we can actually start investigating that. The other thing that comes out, for instance, is that, of course, the silicon retention also depends on the age of the reservoir. Just after closure of the reservoir, retentions tend to be very high, because the diatoms and the other silicious organisms will immediately start taking up the silicon, but it takes some time to start recycling, to start redissolving the silica. So initially we have high retentions, and then very quickly it tends to stabilize. All right, so I'm at the end of my talk here. So some of the messages, the take-home messages here. First of all, it's a fairly straightforward message. Humans are modifying both the absolute and relative nutrient loadings to rivers, with far-reaching implications from individual ecosystems to the global biosphere. But I really think it's important, because there's too many people, too many research that say, I work on nitrogen, I work on phosphorus, and then a few of us, we work on silicon. But in fact, we should work on all these nutrients together, because it's not just the absolute fluxes that are important, but also these nutrient ratios. And the nutrient ratios won't predict to you what's going to happen necessarily in an ecosystem, but it's a primary pressure, or changes in nutrient ratios are one of the primary pressures on aquatic ecosystems. Another fairly obvious take-home message: river damming represents one of the major anthropogenic disturbances of the natural cycles of water and of nutrient elements. Partially to this retention, but also by changing, for instance, carbon exchange fluxes with the atmosphere. Reservoirs play an important regional and global role in the biogeochemical processing and retention of limiting nutrients. I hope I made that point clear in my presentation. And then finally, a mechanistic understanding of in-reservoir processes helps scaling up the relative sparse data on reservoir biogeochemistry to make global scale assessments of the environmental effects of river damming. So I think once we start merging mechanistic understanding with data, of course, we're better off than before. And the last message, of course, more research needs to be done, of course. And so that's it, and I'd like to thank you for your attention. Thank you.
Thank you very much, Philippe. We have time for some questions and discussion. Helen.
This is really the the whole purpose of doing this. So that would be the thing. And we have some colleagues in France who are actually developing river models where they want to represent reservoirs in a more, I would say, in a more realistic way. But the ultimate goal is then to say, okay, what's going to happen at the coastal zone? And so you can then use these ratios to at least make some inferences about what could be potentially the most limiting element then for the coastal zone. Can it explain some of the change we're seeing? Can it, for instance, explain some of the increased occurrences of algal blooms in places like, for instance, in the South China Sea? But that's really what one of the goals is. I mean, it's not the only goal, but that's certainly one of the outcomes that we hope to get or to achieve by applying this type of approach, not only to silicon, but also to phosphorus and to nitrogen. And iron is also an interesting story.
Jeff.
Yeah. It's an excellent question because essentially that's the big unknown. I mean, if it's dissolved silica, we know what it is. It's silicic acid, etc. So that's why I refer to it as reactive particulate silicon. So it's silicon that on a relatively short timescale can potentially become bioavailable. In other words, can dissolve and then can become back to the dissolved state and be taken up by an organism. So there's a variety of different ways that people try to quantify this reactive fraction. Typically, it's by some kind of a chemical extraction, an alkaline chemical extraction. In the literature, very often you will see people referring to this extraction as the biogenic silica. But in fact, this alkaline extraction technique will also extract inorganic amorphous silica. And there's more and more evidence that amorphous silica or very poorly crystalline, even very poorly crystalline aluminosilicates might actually be released by this extraction. But in the end, if you're a diatom, you don't care whether it came from an inorganic amorphous precipitate or it came from another frustral that is dissolving. It is reactive. With that, there's always some level of subjectivity. When do you say something is reactive from is no longer reactive? That, for instance, depends also on the sedimentation rates in the reservoir. If you have very high sedimentation rates, something that would be reactive, it would be sitting there for a long time at the water-sediment interface, might no longer be reactive because it's being buried, and then further transformation will tend to render it very unreactive. Sometimes you have the impression that silica biogeochemistry is simple. It's either dissolved or it's under the SiO2 form. SiO2 recovers a lot of different materials. And in fact, you can take a very amorphous silica gel, let it age over time. It will still be SiO2, but its kinetic properties will be very, very different. And its availability to organisms will be very different. So one of the ideas I have, and I've been trying to push people in my lab to do it, is actually not to try to determine the bioavailable fraction of particulate silicon through a chemical means, but through a biological means. If a diatom can extract it, then it's bioavailable. So to actually use a biological assay to determine what is the bioavailable particulate fraction. But speciation of silica is more complicated than the little formula SiO2 might make us think. By anything that wants, so if you have benthic vegetation that takes up silica, it's all in there. Now, for the parameter values, to get at least some kind of a bulk part value, we use the uptake kinetics of diatoms. But in principle, it can represent biological uptake of silicon by any organism that requires silicon. Yeah. Yeah. I'm a Mac person, so it takes a while. Oh, that's what I wanted. Yes, so this maximum uptake, in the model itself, the uptake is represented by a kind of Michaelis-Menten kinetic. So that's the R max, the maximum uptake rate, but it will be modulated if you don't have enough reactive silica. The actual uptake will be lower. Yeah, sorry. Yes, and so that's right now in this particular version of the model, we cannot do that because it's a silicon-only model. The idea is ultimately to link it to a phosphorus model, to a nitrogen model, so that the system can choose what the most limiting nutrient is. And light. So yeah. Yeah. I would suspect it would. The answer is, I don't know, but if you talk to Taylor, maybe she'll be able to pull out the table of the various reservoirs. It's been an enormous job just to extract some of that information from the literature. I'm sure there's much more. I mean, people do, on a fairly routine basis, measure dissolved silica. It's hardly ever reported. So when I first arrived at Waterloo, somebody pulled out, oh, for the Grand River, here are silica data. Yeah. Where is it published? Oh, we don't publish it. It was not nitrogen or phosphorus. Yeah. 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 Now, you need to know that there will always be some silica. So even if silica is ultimately limiting diatom productivity, there will still be diatoms there. I mean, there's still a certain amount of. It's just that they will actually end reproducing even if there's still excess other nutrients available, and then other organisms will take. So you will still have diatoms. So potentially it can still be used as bioindicators. But yeah, I would be very careful, because what you can also see is that sometimes when silicon becomes limiting, the recycling of the silicon becomes more efficient. So you might be losing more of your diatom records simply because they're redissolving to be reused again. So yeah, I would be very careful. The first thing I would do is to first look at, is there a potential for silicon limitation in the particular reach of the river that I'm interested in? If not, then you're fine. But if it is, then maybe you have to be careful, yeah. And in fact, that's something that's been also known by oceanographers. They use these transfer functions, where they know that what they find in the sediment is no longer what was present in the photic zone, because you've lost a lot of the species, a lot of the biomass, or the biosiliceous mass.
Sort of following up from Jeff's question, so you gave us a startling number, 3/4 of the silicon coming into the Diefenbaker reservoir is retained. So could you speculate on the implications in terms of the downstream biogeochemistry?
Well, the thing is, from what I hear, is there's even more phosphorus retention than silicon retention, relatively speaking. Mm-hmm. So if the system, the short answer is I don't know. It's my short answer to most questions. But if you have water coming in where essentially it's not, silicon is not limiting, and you have more retention of the phosphorus than the silicon, then I would not expect necessarily silicon limitation in the downstream reaches. Mm-hmm. Of course, in a system like this, you of course get additional additions after the dam. Mm-hmm. But if indeed, and that's why we're interested in the relative retentions, because it can tell us whether a system is going to become more phosphorus limited or less phosphorus limited or more silicon limited when we put in a dam. But so from what I understand from the work that's been done here on phosphorus, there's even more phosphorus retention, relatively speaking, or the percentage phosphorus retention is even higher than the silicon retention.
Yeah. Jeff, yeah.
Limits it, yeah. 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 So ultimately what we like to do is also have similar modeling for carbon, where the carbon, the CO2 fixation would have an explicit turbidity limitation, which then would work its way back to the utilization of the other elements. But I completely agree with you. That's something that's missing. So I would not take these, so these are just, so part of the variability that you see, it's not a perfect relationship, that already tells you that this cannot be the single control on the siliceous productivity in the reservoir. It must be other controls. Could be other Newton elements, could be turbidity, could be stratification, could be a lot of different things. This model, I mean, this is a 4-box model that cannot capture all the complexity, of course. So, yeah, 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 yeah. Yeah, and that's why this is the first step. So we start as simple as possible, and then from there, hopefully we'll be able to refine.
Okay, well, I think time is moving on, so I'll call the meeting to a close. I think we've had a really interesting insight from Philippe into perhaps some of the less well-known aspects of nutrient chemistry, salutary story about, of course, the roles of human intervention in the natural environment, which we're still trying to unpick in so many ways, but also I think the intellectual bravery in trying to take our limited understanding and actually make some extrapolations to look at global scale processes. And we've really enjoyed your talk, Philippe, and thank you so much for coming.
Thank you again for inviting me.